doctor 2 years ago, and then I realized that the difference between a very bad prognosis and a very good prognosis has to do in the biology of all of this. And so what ended up happening after that is that I started reading as much as possible. I went online and I read as much as I could, all of the literature that was available. And this went on for a couple of months until I realized that that just wasn't enough. I ended up writing a letter to the American Association for Cancer Research, and they allowed me to come to their annual meeting and to listen to what all of the researchers had to say. And I listened to some of the scientists who were standing on the forefront of cancer research, and I got to listen to all of their different breakthroughs and just where the field was and where everything was going. This was very inspiring for me, and so I came home after that experience and I began writing research proposals for myself to perhaps do someday. Months after that, I had the opportunity to work with Dr. Rhodes, who's a professor at the Florida Gulf Coast University, and she allowed me to come into her lab and actually begin to perform my own research. And this was an absolutely incredible experience because the very next year, I returned to the American Association for Cancer Research and was the youngest person to present at that meeting. After that experience, I came back and I started thinking. I started thinking about all of the possibilities that had to align in order for me to get to the place where I am today as both a cancer researcher and a student and just a member of this academic society. And I started thinking that it's very possible that there are many young girls who have a similar idea or affinity for some sort of STEM field, and they're just lying dormant waiting for that opportunity to come along. And I started to receive letters from young girls who told me that I inspired them and that my story almost lit a spark in them. And because of those girls and because of those letters, I put together this not-for-profit organization called the STEMness Movement, and this is the inaugural workshop. Thank you for coming. I would also like to thank our donors who gave us money to rent this lovely space and to have all of the materials that we have for today's meeting, as well as all of the technology that we have here hooked up. So thank you. So without further ado, we're going to get to the point. So we are all here today to answer the question, why haven't we cured cancer yet? Now, when we think about this question, why haven't we cured cancer yet, really, there are three questions that underlie it. And that is, what is cancer? How do we treat it? And why are the treatments not working? So let's start from the very beginning. What is cancer? So I'm going to ask you, particularly the students, what do you know about cancer? What do you think cancer is? What have you heard about cancer? Anything? Hands? Anything? Okay, high schoolers. Don't make me ask the doctors. Alrighty, Amanda, what do you have?
Cancer is a very serious illness that has to do with unstoppable cell reproduction.
That is absolutely correct. Thank you. Sam, do you have anything to add?
Cancer that starts in one place can travel throughout the body.
Yes, that is true. Metastasis is a huge problem. We're going to talk about that at a length later. Thank you. Anybody else? Alrighty, that is ample information. Thank you. So, I've got this lovely picture here where all of these cartoons are supposed to represent life. So all of life is made up of cells. And if you look, not all cells look the same. So this is just a picture of plant cells, and that's a picture of animal cells. The main takeaway that I wanted everyone to have is that all life is made of cells. So every part of your body, every part of your dog's body, every part of the tree outside's, well, not body, but the tree itself, that is completely composed of cells. And so we're going to talk a little bit about what cells are capable of doing. So cells are capable of communicating with one another. Now this may look somewhat complicated, but the way that this works is that one cell sends out these small bioparticles called ligands. And you can think about those as sort of a message. So one cell sends a text message to another cell to do X thing. And then once that message is sent, it receives that text message, it reads it, and then it does the action that is embedded within that message. So cells are also capable of dividing. So if you look at this picture, you can see that there are different fluorescences shown here. So you've got your red fluorescent protein and your green fluorescent protein present here. So as you look, lovely Jake is doing this over and over, one cell is pulling apart. So the way that it does that is it takes the genetic information and it multiplies it by 2. It replicates that genetic information. And then what the cell ends up doing is it creates these strings almost that take that genetic information and pull it apart. Now that genetic information is stored in the form of something called chromosomes, which is DNA really tightly packed together. So if you look, you can see the process of cells replicating their DNA. So just a second ago, I was talking about how cells need to replicate that genetic information in order to have the blueprints once they become 2 different cells. So you can see all those proteins, all those globular things, and they are working together to create multiple strands, well, to pull apart and to recreate the missing strand of DNA. We will talk at length about DNA later. So cells are also capable of creating proteins. Now, this is known as the central dogma of biology. So what cells do is they take DNA, and from that they create RNA, and then from there, other proteins create it into a protein. But here's the question. Could DNA replication affect the creation of proteins? So in that process that we were looking at earlier, where the DNA was getting pulled apart and then built up again by the proteins, could that affect the creation of proteins in and of itself? Well, a scientist by the name of Robert Weinberg did some calculations, and this is what he found. So he found out that approximately 3.2 times 10 to the 15th mutations in a lifetime are due to only cell division, that DNA replication that naturally occurs. And so if you're not sure what 10 to the 15th is, think about it like this. It's 1,000,000 times 1,000,000 times 1,000. That is a lot of mutations. And so you can think about this in a way that the older you are, the more mutations you're going to have. So do all of these mutations result in cancer? Because that's a lot of mutations. Well, some do, but not all do. Yeah. No, not all. So I'm going to return to the original question, because we just spent a lot of time talking about basic biology. So what is cancer in and of itself? Well, cancer is not a single disease. In fact, it manifests in several different places throughout the body and in several different forms. In fact, there are over 200 types of cancer. But the question is, what do these 200 types that develop in vastly different parts of the body, what do all of those types have in common? The answer lies in a paper called The Hallmarks of Cancer. This revolutionary paper was written by 2 scientists by the name of Hanahan and Weinberg. And this paper talks about all of the similarities, all of the traits that cancerous cells have. So there are 10 main traits. Some of them we will go over at length in comparison to others. And the first one is sustaining proliferative signaling. Now, can I see a show of hands? How many people in here know how to drive a car? So you hit the gas pedal, and the car starts going really, really fast. So similarly, so remember when I was talking about how cells talk to each other? So This proliferative signaling is almost as though the cell steps on its own gas pedal and it starts dividing and growing very, very rapidly. But what it's able to do is create its own signal. It's able to hit its own gas pedal and grow however much it wants. Almost conversely, the next hallmark has to do with evading growth suppressors. So the first question gave it away to some extent, but can someone tell me what the brakes do? Anybody? Really? Yeah. Perfect, thank you. So, similarly, cancer cells have broken brakes. So if you think about it, what ends up happening is that it's going really quickly, and then they don't have brakes, so they can't stop. Most cells have this ability to stop once they're right up against each other, if they're growing a lot. Once they start feeling each other, that interaction causes cells to stop growing. Cancer cells don't care about personal space. They will keep growing no matter what, no matter how tightly packed they are. They're like sardines. They don't care. This next hallmark is actually the basis of one of the fundamental pillars of immunotherapy, which is a type of cancer therapy that we will get at later on. But avoiding immune destruction is a very interesting hallmark of cancer because most diseases can be recognized by the immune system. So your immune system is the part of the body that looks for stuff called pathogens. And those pathogens are what causes you to get sick. So pathogens are foreign things that wind up in your body and make you, well, essentially are trying to take down your body. What your immune system does is it fights back. It gives you that fever. It gives you all of the cells that fight against those pathogens and allow you to continue living. So in terms of avoiding immune destruction, what cancer cells are capable of doing is they're capable of hiding from the immune system so that they go by undetected and so that the immune system does not come and kill them. And it's a very interesting interaction because they're just able to camouflage themselves and to make themselves look as though they are part of your body. I'm not going to do that anymore. I apologize. You can go ahead. So this bounces off of what I believe Amanda said about cancer cells being able to replicate a lot. Enabling replicative immortality has to do with the idea that cancer cells can divide and grow however long that they would like, whereas ordinary cells would simply grow until, well, they realize that they're done for, and then they go through a process called apoptosis, which is cell death. So enabling replicative immortality simply means that cancer cells are able to continue dividing infinitely, however much they would like. The next hallmark is tumor-promoting inflammation. So what this means is that in the tumor microenvironment, which is basically the section of your body that is completely infiltrated by cancer. So this section of the body looks very similar to an inflamed area. And the reason why this is important is because this allows that area to get the nutrients that it needs in order to continue growing at such a rapid rate. This is activating invasion and metastasis. So like what Sam said earlier, cancer cells are notorious for seeping into other parts of the body and just invading. And this goes into the next hallmark, which is inducing angiogenesis. Now that's a big word, but this is what it means. It means that cancer cells are capable of manipulating the surrounding environment in order to create their own blood vessels. This is important because this is what causes metastasis. Once cancer cells start having their own blood pathways, they can get the nutrients that they need in order to continue growing. They can get oxygen and they can get food, and both of which are very important for the survival of cells. They're also able to spread to other parts of the body, which is one of the factors that makes this disease so lethal. This is just another trait that cancer has. Genomic instability is another hallmark of cancer that has to do with the fact that it's dividing incredibly rapidly. And in order to do so, it needs to replicate its own DNA extremely quickly. And this leads to some mistakes. Because, you know, when you rush your work, you have a tendency to make mistakes. Cells are exactly the same. So as these cells are growing and dividing and changing very rapidly, they make these mistakes, and it results in an unstable genome. So every living thing, just as it's made up of cells, it also has its own genome. So humans have their own genome, and just as corn has their own genome, just as bacterium has their own genome. Cancer cells have an unstable genome, meaning that their genetic information is just very messed up. And it is just flooded with all of these different mutations that cause different things to happen within their cellular environment. Resisting cell death. So ordinarily, there are these checkpoints almost. So as cells live and thrive and grow, they end up dying at some point, as we all do. But cancer cells don't have those checkpoints, or they ignore them, basically. So rather than dying, it will continue renewing itself and continue proliferating and growing, and that's pretty much what this means. And this last hallmark is deregulating cellular energetics, which basically means that cancer cells acquire their energy in different ways. So, again, they're growing very, very quickly, and they're living their lives at a very rapid rate. And this means that they require more energy than the average cell. So this makes sense in that they acquire that energy through a different way.